Hello and welcome to a quick review for Anatomy and Physiology Test 3 online. What I'm going to do here is I'm going to run through a few things and just talk you through how the test is going to be structured and some of the main important bits that you might want to consider looking over. Consider this what I would study if I were you. Now, the first comment I have to make on this test is that people tend to do well on it because of the way in which it is structured. All right? The first half of the exam, all right, the first full half of the test is basically a big sequence. One thing leads to the very next thing, leads to the next thing, leads to the next thing. It all leads together in a fashion that makes it pretty much a paragraph that you're having to assert the correct answers into. And mind you, this is a lecture test, so it's all multiple choice. It's all multiple choice. But very much so, everything leads from one to the next, to the next, to the next. There's little real chance of you getting off sequence and missing a lot of things. If you have a decent, fair grasp of what you're talking about, you should get the first half of the test correct with no, or with less effort than would otherwise be required. So with that in mind, let's talk through what I'm asking you in this first half of this exam, and then we'll move on to sort of the general questions. All right. You can guarantee that on this exam, I will be providing you with some sort of stimulus, be that hearing something or seeing something or touching something, some sort of stimulus, and you're going to have to tell me sort of where in the brain that stimulus would be interpreted. So you need to know about the lobes of the brain and what those lobes are sort of responsible for, what the cerebrum does versus what the cerebellum does, the role of the hypothalamus and the thalamus in this. Like, you need to walk me into the brain using sensory receptors and then walk me through the brain as to what would be doing what there. You can most assuredly guess here that you will be making a decision to then do something, and that something you'll have to do is guaranteed to be some sort of muscle flexion. So in the world of muscle flexion and sensory reception, you need to be able to tell me about terms like visceral sensory, somatic sensory, visceral motor, and somatic motor. Remember, visceral means organ, somatic means of the body. Viscera we are not always aware of consciously, whereas somatic we are consciously aware of it. So like a somatic motor output would be me grabbing my pen. A visceral motor output might be my hypothalamus releasing a hormone or something like that. See the difference? Like, things are being done, but one I'm consciously aware of and the other happens behind the scenes. This is the way you have to approach this situation. Now, when you send a stimulus out from the brain to lead the muscle flexion in this first half of the test, you need to be able to walk me through parts of a neuron. You need to be able to tell me about how neurons have dendrites that receive messages, a nerve cell body that processes information, and then an axon that sends action potentials. So the nerve cell body, the dendrites, they receive what are called graded potentials. Graded potentials are decremental. They get weaker over time. But if the graded potential reaches what we call the trigger zone or the axon hillock, that's the area where the axon connects to the nerve cell body, it will fire off an action potential that will shoot down the length of that neuron. Now, action potentials. You need to be able to walk me through the wave of depolarization, the wave of repolarization, the brief period of hyperpolarization, and return to a resting state. I want to know what's happening during each of those phases. So you got this action potential. It's traveling down a neuron. When that dyslexia is hard, folks. When that action potential reaches a very specific spot, you'll have a wave of depolarization, followed by a wave of repolarization, etc. I want to know what ions are exchanging place there. Now, further, you need to be able to walk me through the role of myelin in your peripheral nervous system. So, and for that matter, you need to be able to tell me about myelin in your central nervous system too. But in the central nervous system, that question is mainly talking about gray matter versus white matter. Whereas in the peripheral nervous system, I'm wanting to know about how we speed up nervous transmissions. So, How can I make a nerve transmission go faster? Tell me about the diameter of the neuron. Tell me about having myelin or not. Tell me about the role of Schwann cells. Tell me about how in the peripheral nervous system, if Schwann cells are present, you have an increased likelihood of healing a damaged neuron. Okay, all of those things are important. All those things. For that matter, tell me the difference between in a myelinated neuron, you have what's called saltatory conduction using the nodes of Ranvier, versus in a non-myelinated neuron, you have continuous conduction. That's where the stimulus kind of travels down the neuron in a continuous fashion. It just runs down the whole length. So be able to tell me the difference between those. How do you make that neuron go faster? And where in the body would you expect to see myelin versus not myelin? Now, when we reach the end of the axon, you've got to walk me through the axon terminal, synaptic cleft, and motor end plate. I want to know about how calcium goes into the axon terminal. I want to know about how the axon terminal releases acetylcholine. That's a neurotransmitter. I want to know how acetylcholine binds to the motor end plate and causes the influx of sodium into the motor end plate and potassium out of the motor end plate. I want to know about the depolarization wave that starts. It's called an end plate potential. How that end plate potential spreads down the length of the muscle fiber. How it travels in deep to the muscle fiber using the T-tubules. How it stimulates the terminal cisternae of the sarcoplasmic reticulum to emit calcium. That calcium that binds to troponin, which pulls tropomyosin out of the way, revealing actin's binding sites, so that myosin can grab a hold of actin and pull. Okay, so these are the myofibrils. You got to take me from the nerve all the way into the myofibrils, and then walk me through what's called the cross bridge cycle. Okay, there's a slide in particular on it. So I want to know about how the sarcoplasmic reticulum stores, calsequestrin. I want to know about how calcium is released. I want to know about how that calcium binds to troponin. I want to know about how it pulls tropomyosin out of the way, how myosin is then capable of binding to actin, and I want to know about the cross-bridge cycle and the role of ATP both in muscle flexion and in muscle relaxation. All right, and that's muscle flexion right there in a nutshell, folks. That's muscle flexion in a nutshell. All right, so that's basically the first half of the test. That, however many minutes this has been so far, 7 minutes. If you understand that 7 minutes of material, you're doing pretty good on the first half of this test. Okay, I would say that knowing 7 minutes of material for half a test is not bad. So that's progress. All right, then we've got a few other things that we need to talk about. You need to know about how we protect the brain. So tell me about the meninges. That'd be dura, arachnoid, and pia mater and the roles that they play. Tell me about cerebrospinal fluid and how it sort of helps float the brain, how it provides almost like a friction barrier, how it provides a means of material to flow around the brain and carry hormones or what have you. All right. Tell me about the blood-brain barrier and how this sort of prevents certain chemicals from being able to get into the brain, and how it lets other things through quite readily, and the role of the blood-brain barrier in protecting the brain. All right. Tell me about, let's see, I think I already mentioned white matter and gray matter in the brain, but tell me what gray matter is. Tell me what white matter is. I want to know about it. Do you only find gray matter on the outside of the brain? No, that's in basal nuclei, deep to the core of the brain as well. Yeah, man, absolutely. You can guarantee that you're going to have to know the major functions of the hypothalamus, the major functions of the thalamus, the major functions of the cerebellum. All right? I will ask you about, I don't know, what controls your general motor activities when you're walking around from place to place. That's your cerebellum. All right, so you need to understand this and have a good grasp of it. All right, we had several degenerative brain disorders that it's important for you as a functioning human being to be able to recognize, just so you can see it and then address problems sooner rather than later. So be able to walk me through the major characteristics of the degenerative brain disorders that we've talked about. Alright, let's see what else we've got here. Looking at my notes, give me a minute. Serial processing versus parallel processing. Tell me about the sympathetic division versus the parasympathetic division. Tell me about the different types of muscle fiber. So why does a bodybuilder look super huge and bulky? What kind of muscle fiber predominates in their system? Why would a runner or a swimmer have a slimmer build yet still be in amazing shape? Why are their muscles smaller than would a bodybuilder's muscles be? What type of musculature predominates? And what does it mean about the efficiency of their muscles versus the general strength of their muscles? And be able to use the term hypertrophy or hypertrophy. Tell me about how neurons are capable of sending materials up and down their axon. So if you need some sort of protein or enzyme, generally speaking, at the end of an axon, it has to be made in the nerve cell body because that's where the nucleus is. I want to know about how they send things down the axon and how they send things back up the axon. There are two special words there that you need to be friends with. So go and take a look at that. Tell me about the neuroglial cells. I want to know about their names and what they do. Who are the neuroglial cells? Give me their names. What do they do? Tell me about... hang on. Reading here, folks. Be friends with that action potential. Be friends with the knowledge that it's an all or nothing response. Tell me about what ions are exchanging place during the different phases of an action potential. There are several parts of the limbic system that are very important for our general daily lives. I would like for you to be able to walk me through the limbic system and just sort of tell me what does what. Okay? These parts deal with our emotional state, deal with our memories. Like, they really help us do a lot of things in our daily lives. And I think that you need to have a familiarity with the limbic system. So just be able to walk me through it. Tell me about handedness, left-handedness, and right-handedness, and the role that might play in cerebral lateralization, and sort of what side of the brain does what. So how would a person that's left brain dominant behave generally versus a person that's right brain dominant? What might their skill set be? Tell me about muscle coverings. Tell me about how they carry in blood vessels. And generally speaking, tell me how muscles are structured. So how they connect to bones, generally speaking, being fleshy and or via indirect attachments. And tell me about what an origin is and what an insertion is. Just sort of how the muscle is structured in the body to lead to the ability for flexion. And folks, I'm going to say that is a really nice review for this exam, and I hope you do astoundingly well. Thanks.